Molecular biology and physical techniques are at the core of any biochemistry interview. It's absolutely essential to prepare to talk about and understand these techniques. Hi everyone, my name's Elvi. I'm an Oxford biochemistry graduate and the founder of Elvis Oxford Coach, a university admissions coaching service for Oxford and Cambridge for the bioscience subjects. In this video, we're going to do some Oxford biochemistry interview practice. The focus of the question will be on proteins and how to study them experimentally. This video will cover explanations and applications for a wide range of biophysical and molecular biology techniques that are commonly used in biochemistry. This knowledge will be really applicable to a broad range of questions that you could possibly get in your interview. You'll want to stick to the end of this video because we go through a lot of information in quite a short space of time. Proteins are at the heart of investigation in biochemistry. Proteins are what give cells function. But how, as a biochemist, can you produce a protein in order to have enough quantity to be able to study it? This is what we're going to look at now. To solve this problem, we're going to go through it step by step, which is exactly the kind of thinking strategy you need to use to be successful in your Oxford or Cambridge interview. The central idea to this problem is that you want to think of cells as protein factories. Don't think of making proteins as you would synthesizing something in your organic chemistry class, okay? It's much more cost efficient to get a cell to make a protein for you. So how do cells make proteins? Well, from DNA, right? DNA using the cellular machinery. Logically, therefore, the first step is to make the DNA that the cell needs in order to make the protein you want to study, okay? And this is usually done with the polymerase chain reaction technique. The end product of a PCR process is that you've produced the specific DNA of interest at a very, very high quantity, which is absolutely necessary if in the end you want to achieve or have the potential to achieve a large amount of protein output. Why? Because you're going to be using a whole colony of cells to grow this protein in sufficient quantity. Therefore, you need lots of DNA to put into those cells. So the next step is actually getting that amplified DNA into the cell. And this is usually done with a vector. So on the slide, I've given you an example of one type of a vector known as a plasmid. Now, this plasmid has multiple features, some of which we'll discuss later, okay? But the one I want you to focus on right now is this multiple cloning site. So this is a region of the plasmid which can have DNA inserted into it. How can this be done? Well, this multiple cloning site has lots of sequences which can be cut by restriction enzymes, okay? And so using the restriction enzymes, you can cut open the plasmid, and using the same restriction enzymes, you can cut the ends of that DNA fragment that you've produced, you've amplified using the PCR process, and fit it into the plasmid, okay? So one thing that I want you guys to think about is, why do we need a vector, okay? Why is just inserting the DNA fragment on its own not going to work? Think about what could happen to that DNA inside the cell versus a structure like this, which is a plasmid. The DNA is in the plasmid and the plasmid is in the cell. How do we get the protein from this DNA? Okay, well, as I said, these plasmids, they have some specific features. And one key feature that I want to highlight is an inducible promoter. What does this mean? As we know, a promoter is necessary for starting the transcriptional process. It's where RNA polymerase binds. The difference is, in this promoter, compared to all the promoters in all of the other genes in the host cell, i.e. the bacterial cell that we're using to make this protein, this promoter is under our control as the experimenter. It's not under the complex regulation of transcription as per the cell is under our control. How does this work? Well, these plasmids can be engineered to contain promoters that are specifically activated simply by the addition of a chemical, right? So imagine this, you have all your cells in a broth. These cells, they contain the plasmid, okay? And your protein's not being made until you add the chemical to this broth, and then suddenly only this promoter, and that's the key thing here, only this promoter is going to be active when you add that chemical. So what is the outcome? A huge amount of the protein is produced, which is very important because in order to study a protein, we need to have a sufficient quantity of it. The next step in the process is to actually purify your protein. What do I mean by this? We're using a cell to make this protein. In addition to our plasmid, which contains the gene of interest that we're after, this cell also has its own genome. It's going to be making hundreds and thousands of other proteins. So how do we separate our protein of interest from all the other proteins that that cell is going to make? One way to do this is to include a specific purification tag in the plasmid that we're using to make our protein of interest. As I said, these plasmids have lots of features, and one of them is a his tag. What does that mean? So his stands for histidine, and histidine is an amino acid. Now, this histidine tag is actually a six-fold repeat of that histidine amino acid. So if it's an amino acid, what does that mean? It means that obviously this amino acid can be encoded for genetically. So this his tag is just an extension to the DNA that encodes for our protein. It's just a series of codon repeats that give rise to a sequence of 6 histidines in a row. Why is this important? How does this help us with purification? Two reasons, okay? The first is that this amino acid sequence doesn't appear in nature, so it's very specific to this system. The second feature is that histidine has a strong binding affinity for nickel. We can exploit this, okay? And this is what enables us to purify the protein. So let's take a look at how this works. You finish the reaction, you've induced the promoter, and you've taken the reaction to completion. Now you have a bunch of cells in a broth. You lyse the cells, and then you obtain a solution containing all the different proteins of the cell in addition to your protein of interest. Okay, what you do is you pass that solution on top through a column. Now, this column contains particles, beads, plastic beads covered in nickel ions. Now, only the protein of interest, which has this his tag, is going to have that strong interaction with the beads. Therefore, all the other proteins are going to pass through, but your protein of interest remains on the beads. Therefore, you have separated, you have purified your protein of interest from everything else. Now, the final step in this process, which is very important but often overlooked, is that of quality control, i.e., checking that you've actually obtained enough of your protein of interest. Okay, so what's the best way we can do this? The most specific way, the most reliable way to detect a specific protein of interest is using an antibody. Okay, the antibody antigen interaction is the highest affinity interaction known in biology. Essentially, the way this technique works is that you take your protein of interest in a solution, and you essentially wash on top of it a solution of antibodies against that protein. Now, what you would expect is that there is a binding, okay? And then what happens is that you use a secondary antibody against that first antibody, and then that secondary antibody has some kind of detection mechanism. Either it's bound to an enzyme, and then that converts a substrate into a convertible product, or, for example, the antibody is linked to a fluorophore or a radioactive molecule. But the key principle is that if you don't actually have your protein in sufficient quantity, or if it's not there at all, you're either going to get a very low response on this test, or you're going to get no response. Let's briefly just recap all the techniques that we've looked at so far through this question. We've talked about plasmid vectors. We've talked about PCR as a way to amplify a bit of specific DNA. We've talked about the concept of an inducible promoter, i.e., a promoter that is under the experimenter's and not the cell's control. We've talked about a Western blot, which is a way of detecting a specific protein of interest, confirming its presence using an antibody. And we've also talked about column-based affinity tag purification. So that was using the 6-histidine tag built into our plasmid vector as a means of purifying that protein. Now that we've purified this protein, or let's say we've been handed an unknown purified protein, what are some of the things that we would want to know about this protein, and how experimentally could we investigate this? So we'll take a look at a brief summary of some of the most important things that you'd probably want to know about a protein. So an obvious thing is size. How big is it? How can we determine this? Mass spectrometry is one way. We can determine the approximate molar mass, and using bioinformatics, we can calculate, we can predict likely candidates that correspond to that molecular mass based on the DNA sequence. Another thing we might want to investigate are the protein's interaction partners, and we can do this using techniques similar to what we've already looked at, i.e. the column-based affinity purification method. Instead of having a column full of beads coated in nickel, we can have the beads be coated in our protein of interest. And so to determine the interaction partners, all we would do is we would take a cell lysate, i.e. the cytoplasmic contents of a cell in which all the proteins are going to be dissolved. We're going to take that solution, pour it onto the column, and then see which proteins bind to our protein of interest. One of the important things that we would want to investigate about a protein would be its function. And so in biology and biochemistry, when we talk about investigating a protein's function, ultimately what we're going to be doing are genetic experiments, i.e. adding that gene in, removing that gene in, interfering with its transcription or translation, and seeing what happens to the cell. That is the rigorous way to determine the function of a gene, the function of a protein. Another thing that could be of interest to us is, where is it in the cell? So in order to do that, we need to see the protein. But how can we see a protein, right? We can't just put a cell under a microscope and visualize every single protein there. Visible light doesn't have the right resolution. But what other approaches could we use? Potentially, we could use fluorescence. And in the same way that in our plasmid we had a tag for histidine, the plasmid can incorporate a tag for a green fluorescent protein. This green fluorescent protein would be attached to our cell, and therefore we can see this, the protein moving around in the cell. That's one approach. At the heart of all of this really is structure, right? The structure of the protein is going to be related to the size. The structure is going to determine the function. The structure is going to determine the interaction partners. And to a certain extent, the structure, the type of folds that protein has, the hydrophobic versus hydrophilic regions that a protein has, is going to determine where it is in the cell. And some of the methods that we can use to do that include X-ray crystallography or NMR. Again, it depends on the type of protein. But this is the general idea. These are just some of the most important things that you can mention, you need to be thinking about when studying a protein. Molecular biology and physical techniques are at the core of any biochemistry interview, whether you've mentioned them on your personal statement or not. It's absolutely essential to prepare to talk about and understand these techniques. I really hope you found this video informative, and in addition, I hope it really emphasized the importance of not only having biochemical knowledge, but also really understanding the importance of experimental techniques and knowing how to apply them. If you feel like you need further personalized support to help you prepare for your interview, you can go to my website, www.elvisoxfordcoach.com, and you can book a free consultation with me to discuss how we can best work together in order to maximize your chances of success at interview. If you book a session with me, what you can expect is a 1 hour long mock interview tailored around your specific personal statement with detailed discussion and explanation. This is obviously a stark contrast to the real Oxford interview, where you only get 20 minutes of interview time and often little to no explanation is provided. Furthermore, I offer full verbal and written feedback. And alternatively to the mock interviews, we can simply do taught lessons very much in a more expanded form of what I've done here. And these can cover a wide range of topics. It can be the central dogma, it can be a techniques based lesson, so like what we did here, energy and biochemistry, organic chemistry, or working on interpreting scientific articles. Thank you very much for watching. I hope you enjoyed it and I hope you learned something. Let me know in the comments what you most enjoyed about this video and what you'd like to see in upcoming videos. I'm probably going to do 1 or 2 more interview preparation videos before the December interviews start taking place. So please do let me know what you'd like to see. As always, I wish you all the best with your Oxford and Cambridge applications. Thank you.